Dr. Sabarinathan from NCBS, who is finding his way to the podium. Thanks, Coach.
And other organizers for this opportunity. Myself, Sabari. I started my research group at NCBS 4 years back, and our lab is broadly interested in understanding cancer evolution through genomics. As we have heard from first speaker and also from Karthik before, we know a lot about cancer. We know how it develops and how it progresses, and we know about the treatment options as well. But still, we cannot completely cure the cancer. One of the problems is it may either reoccur or it will develop resistance to the treatment. Why it is happening? Most of the time it happens because it continuously evolves. And the evolution is being fueled by genetic and epigenetic changes that is constantly happening in the cancer genome. As when they progress under some stress condition, they try to find a new clones and then they will further evolve. And this is the way the tumor kind of recur, or they kind of move to a different sites of the organ, and they can persist and they keep coming. So our lab is broadly interested in understanding how these genetic and epigenetic changes is influencing this cancer evolution. How are we addressing this? We tap on to the data that is publicly available, which could be of different categories. And from this, we are trying to extract information such as mutational processes, which is basically process that creates somatic mutation in the cancer genome. As Dr. Ramesh mentioned in the first talk, there are some variants that we inherit from the parents, and there are some mutations that we accumulate as we go through our lifetime. And most of those mutations are called somatic mutation, and that we acquire due to the exposure to different things. And by looking at the genomes, we can have a fair understanding of where this mutation occurs. And as Karthik mentioned, the sequence context can give an indication of where you expect to see some driver mutations. So those kind of information we extract from this sequencing data, and we use that information to predict cancer driver, which Karthik has covered in greater detail. And we are also interested in understanding how this cancer been shaped by the immune interaction. So as the cancer starts and progress, there is always checks in our body to keep control on that. And that is mainly influenced by the immune cells. And tumor has somehow evolved in a way to escape from these immune attacks. Sometimes these are called as immune evasion, as tumor evolves to escape from the immune processes. And we are also interested in understanding how some tumors evolved over this immune attack. We try to understand this feature so that we can eventually predict or assign particular driver events in each of these individuals, and then by correlating with their clinical feature, can we somehow say that some of these events are biomarker to say upfront who will prognose to a particular treatment or not. So this is what we are eventually looking at. So for today's talk, I will not be talking about the drivers that are caused by mutations, which could be any of these mutational processes. Rather, I will focus on another kind of event that could happen. For example, there are a lot of virus and microbes that are interact with the tumor. In case of cervical cancer, there has been a long association between the human papillomavirus, which is the main driver of majority of the cervical cancer. So in this talk, I will focus on how HPV would drive the tumor progression through several mechanisms. And this work was done by Anurag, who is a PhD student in the lab. Just to give you the context, so we have this cervix layer, and if the HPV gets infected, it's a human papillomavirus, it's a DNA virus of size around 7 kb. Usually it mostly affects the mucosal layer, and most of the time our immune system is good enough to identify this virus and remove it from the body. But in unfortunate situation, if there is any scar, this virus may get entered to the basal layer, which is the layer where a stem-like cells are present. Meaning, if this, usually when this cells in the top layer get old, this basal layer help them to repair these cells or reproduce these cells. When virus enters this basal layer, it takes advantage of this differentiation property, and it replicates its own DNA and produce more viruses. So now it actually hijacks our own body cell to reproduce this virus, and now the virus go and affect more cells. And this was primarily driven by this protein like E6 and E7 that is produced from the virus. And these proteins are long known to be oncogenes because they impair the p53 and RB pathways of these cells. If these two would not have been there, then these cells would not progress. This is the initial stage where the virus exists as an episome, meaning that the viral DNA will be present in the nucleus most of the time or in the cytoplasm. But what happens is that over the time of replication, because of this E6 and E7 activity against the p53, we can have a lot of DNA breaks. And sometimes during the repair, this viral genome can get integrated into the host nuclear genome. Okay? So this is how the viral integration can happen. For this to happen, you need a break in this DNA, in the viral DNA, and you also have to have a break in the nuclear genome as well. And this is very common during the time of DNA replication. And some of the sequence similarity here will make them to use as a complementary stand, and then you get this integration. Now the question is, when this integration happens, how does it perturb the actual function of the human nuclear genome? Because it is integrating to somewhere, right? It could be integrating to a protein coding gene, or it could be integrating to some other non-coding regulatory region. So when it happens, how does it affect the function? So that's the broader question we are asking. So most of the time, the break happens at the E2 gene, which in principle controls the expression of E6 and E7. But when it gets perturbed, this E6 and E7 will overexpress so much than it used to be in the episomal form. In addition, this integrated region also has some local LCR region, control region, which can act as the enhancer or promoter for these genes. So how do we find where the integrations are in the human genome? Thanks to the next generation sequencing, if you are generating the short read sequencing from the tumor samples, which have a HPV integration, you would get some reads that are directly coming from the integrated DNA, since these are short reads, and the integrated region is usually around 5 kb. And you will also get some reads that are spanning both the host as well as the virus. So by mapping back, by using the split read, which Ramesh has mentioned in the first talk, we will have an idea of where in the nuclear genome the DNA comes from and where in the HPV the DNA comes from. So by looking at this together, we can have an idea of in which part of the human chromosome the integration has happened. Okay? So once we have this information, we can go and ask how this integration is affecting the host gene. So as we have seen in Swetha's talk, that any mutations or any event that is happening close to the gene will have an effect on the expression of the nearby gene, which is called EQTLs. Similarly, people have found out that the integrations closer to the gene tend to overexpress the gene very nearby. And this was shown for the MEK as well as for some other cancer genes. But what we are interested is in a broader picture, besides the proximal, can the integration affect the gene in the large scale? Because we know that our genome is organized into several functional layers. So at the bottom level, we have this chromosome being structured like this, and two distal regions can be brought together with the help of CTCF cohesin. You form a nice loop-like structure, and the genes within that are tightly regulated. So we have this kind of DNA loop structures, and there are some chromatin marks specific to this loop. So you can segment the genome into an active or inactive chromatin, and you can also see the chromatin interactions within them. Okay? So this is what's happening in the nuclear genome in general. And now I have this 5 kb region that is a foreign in nature gets integrated to this host genome. So how does this is going to perturb at all this layer is what we are interested. So to check how much this integration is affecting all these layers, we have taken the integration data from TCGA and other cohorts that was detected by using the sequencing-based approach, which I have mentioned earlier. And then we got this chromatin information from the cell lines, because there is not much of chromatin information from the tumor itself. So we have taken the cell line which is closest to the cervical tumor. In this case, it's the HeLa. And we have used normal carotenoid as an additional control. So first we asked how these integrations are distributed in the genome with respect to the different chromatin annotations. interestingly, what we found is that so the darker color is the frequency of integrations that we observed in the normal genome, and then the lighter one is the expected. The expected was counted based on the genomic context where they have integrated. So you can have a rough idea of, given the sequence context, where you would expect more integrations. And we expect that there is a lot of integration can happen in the inactive regions. So inactive regions generally have more repeats, and those regions tend to break during the replication. And this site could probably be the site for integration. And as expected, we also observe a fair number of integrations in the cancer samples as well. But in case of active region, which is the region which are transcriptionally active, we expect very low integration because the sequence context is not that much enriched, but we still see that the observed integration is far more than the expected, meaning the chances of integration in the active region is very rare, but still we see that event recurrently across multiple cancer samples, suggesting that some of this event could be positively selected. Like, it happens rarely, but it kind of positively selected to be present in the tumors. So then we asked whether the integration would affect the expression of the nearby region. So in principle, we know that if a region is inactive, the expression level is quite low. But now the integration has happened, how does that change the expression level of the neighboring sites? So what you are looking here is the integrations that are classified according to the annotation, whether it falls in the inactive region or active region. On the y-axis, what you are looking at is the expression level of genes within 10 kb region. We are looking at a very proximal level, and you could see that both in the active and inactive, there seem to be some level of expression going on in the genes that are neighboring to the integration. So how big is this expression level? And for that, we try to compare it to the control samples. The control samples are again tumor samples which do not have integrations in the same regions where we have observed integration in the other samples. By looking at this, we can see that the samples that have integration have very high expression in the neighboring regions as compared to the controls, both in case of active and inactive regions. And in the inactive region, we could see that some regions are even very much highly amplified to have more expression as compared to the control samples. So this indicates that integration has a potential to change the expression in the nearby regions. Okay, so this we looked at only at 10 kb region, but how far we can take the genomic region to be considered? Because most of the time it is an arbitrary distance. I can take the proximal to be 2.5 kb or 10 kb or 100 kb. But instead of trying out with the different cutoff, we can use this TAD level information. These are all topologically associated domain, where most of the activity are confined to within the same domains at the 3D space. So we have taken this information to demarcate the boundaries and then ask how the HPV mediated expression change are happening within the TAD. So for this, we are trying to look at the expression change in the TAD which have integration. These TADs are usually at several kilobases. And we are also looking at the TAD that are upstream and also downstream to see whether the integration could span the expression change across TADs or at the larger scale. So first we look at the integration in the TAD region, and as we have seen previously, the samples which have integration have a very high expression in the neighborhood as compared to the control samples. But what happens to the neighboring TAD? To our surprise, we have seen that the neighboring TADs are not being affected so much, meaning the genes in the neighboring TAD are not influenced by the integration that happened in this TAD, suggesting that whatever the insulation that is happening at the TAD boundary is somehow preventing the influence of this HPV integration to the neighboring TAD, which is a good news that my integration is not changing the global transcriptional landscape, rather it is confining to the very local region. So we wanted to ask where these integrations are happening at the TAD level and whether the same region is being affected across multiple individuals. So what you are seeing is the different TAD regions on the x-axis. Y-axis is the different patients, and here the colored ones indicates whether the patient has integration in that particular TAD region. And we could see that in majority of the samples, the integration happens at a very specific tag, and we could see also a nice mutual exclusivity too, meaning the sample will not have integration in more than one TAD region. And when we looked at the oncogenes, as Karthik mentioned, there are some oncogenes and tumor suppressor. We tried to look for those genes in this TAD region, and we could find that the one that has the topmost frequent integration had MYC and PVT in it. Okay. So for the sake, I will focus on that particular tag, and this is the MYC and PVT1 within the tag, and we could see that the sample have integration in that tag has a high expression of MYC and PVT1 as compared to the control samples. Then we are interested in looking at where the integration with respect to the MYC gene. Is it happening within the MYC or proximal or at the distal region? So this is the position along the chromosome. The 0 corresponds to the MYC region, and then the points indicates how far the integration is from the genic region. We could see that there are some integration happen directly at the MYC, some at the promoter, and some at the very distal region. But we have seen that all of this cause a high expression of the MYC since they have an integration in this tag. So we try to get to a mechanism to it. So in case of integration that is happening closer to the gene, the way that could overexpress the downstream gene is due to the RNA polymerase. There is something called polymerase runover. It starts with the viral gene, and the polymerase will run to the downstream gene as well. So this way, the downstream gene gets overexpressed as well. But what happens to the integrations that are far away? Like in this case, the integration that we see in these samples are 500 kb away. How does that can affect the expression of the gene that is that far distance? One possible mechanism is through the looping. As we have seen in the nuclear genome, the enhancer and promoter that are far away can be brought together to enhance the expression of the downstream gene. A similar principle can be assumed here. This viral integration also has some regulatory regions. This can potentially interact with the downstream gene through the looping and thereby express the MYC in these samples. Can we test this hypothesis? Fortunately, we can because in the cervical cancer cell line also had a HPV integration in the chromosome 8, which is 500 kb away from the MYC region. So we have taken Hi-C data from this, and we have analyzed this with the help of Gianni. And what we have tried to look here is we have the HPV genome, and then we have the nuclear genome, and then we are trying to understand how much of the 3D chromatin interaction is happening between the viral integration and the host genome. And we could see that much of the viral integration is interacting with itself to some extent, and much of the interactions are going with chromosome 8 in the nuclear genome as compared to other chromosomes. So chromosome 8 is where the integration has happened. Even if you try to localize within the chromosome where the interactions are happening, it is mostly happening in the TAD, which had the integration and the MYC in it. And the interaction is much stronger as compared to neighboring regions, as we have seen previously. So we wanted to ask whether this is happening by chance because of the local distance. This is the interaction that is happening within the TAD, and this is the interaction that is going outside the TAD, which is very less. But if we take this distance into account and then ask how strong the interactions are randomly from the nuclear genome, it shows very less frequency, suggesting that the distance is not an issue. It is mainly the regulation that is mediating the high interactions by integrated HPV. So we went on to test whether which region within the TAD with integration is interacting more, and we could find that the MYC promoter and PVT1 promoter are interacting with the integrated HPV a lot. So this gives a clue of where to look for these integrations and the interactions mediated by this. So we went to the HeLa cell line again, and then we have performed 4C experiment with the help of Dimple Notani at NCBS. So the 4C experiment basically tried to identify the interactions between a region of interest to all other regions in the genome. So we have designed primers for the HPV 18 regions that is in the HeLa cell line, and asked what other DNA regions are interacting with this integrated HPV 18. And we could see that there is a lot of interaction close by, and at the far distance we could see MYC and PBT promoter regions, which we have seen in the Hi-C data, is being very well recapitulated in the 4C experiment as well, which is one more evidence to show that there is a chromatin interaction happening between the HPV-18 and the promoter regions. So if there is an interaction happening, then an outcome of this could be an overexpression of the gene. We have checked the overexpression of the MYC and PBT1 in this context, and we do this by comparing to the other allele which do not have integration. So most of our chromosomes have autosomes. We have 2 copies, and in the HeLa, the integration happened only in one of the 2 autosomes. So we are using the other chromosome 8 as a reference, and then asked how much the expression coming from the integrated copy, and we could see very much the strong allele-specific overexpression of MYC and PBT1 from the HPV integrated copy, suggesting that there is a looping followed by overexpression of these oncogenes. So this is a model that we are proposing from this study. So at the initial stage of the infection, most of the time the HPV get cleared off, but in unfortunate situation it gets entered to the cells, and if it is in the basal cell, they would propagate more, and it will present as episome, like the DNA is staying separate as compared to the nuclear DNA. But over the course of cancer evolution or the progression, this HPV DNA gets integrated to the nuclear DNA, and depends on the location, it may have a direct effect on the nearby gene, or you can have a long distance effect. So in this case, we are showing that the integrated HPV can affect the oncogenes that are far away through the chromatin looping followed by overexpression. All these things were done by using the data that is publicly available from TCGA, which has a strong overrepresentation of the European population. So we wanted to test this in the Indian context as well. Even in India also, 70 to 80% of the cervical cancer is caused by HPV. So we are started collaborating with Kasturibai Medical College with Dr. Amar Rao. So we have recruited very few samples so far. We have 5 samples. Out of the samples that we have recruited, 5 of them have integrations, and all of these integrations were validated by Sanger sequencing. Then we try, by using the TAD approach, we are trying to understand where the integrations are and which genes are affected by that integration. And we got some very preliminary result that one of the gene like XCL8 is being affected, like overexpressed in 2 individuals which have integration at the same location. So this is quite an interesting thing to follow up. But one of the challenges is so far we have been using only the short read sequencing. So we don't know whether is there any other DNA alteration that happened that could also influence the overexpression of the downstream gene. For example, when the integration happens, there could be a local copy number change or local structural variant that could also contribute to the expression of the downstream gene. So hoping that we will use some of the nanopore sequencing data here to resolve the haplotype, perhaps in collaboration with Chirag, to get the haplotype resolved, the integration along with the nuclear genome, so that we can better estimate how much is the effect coming from the integration in addition to the local copy number changes. Okay, with this I would like to acknowledge my group, especially Anurag for all the effort, and Dimple Johnny for the collaboration, and Ramarao for the ongoing work, NCBS and India Alliance for funding, ICGC and TCGA for making the data public, and thanks to you all for your patience.
Talk. So I have a question regarding the basal cells. You said that when the virus infects these cells, that's when it progresses more towards cancer, right? So these TADs that you've identified, are they integral to the function of these basal cells? And if you could block them somehow, would you prevent the progression of this cancer or occurrence of it?
Yeah, very good question. So we don't have the TAD level information from that specific cell type, but the cervical cancer cell line is pretty much close to this one. So even if you look at the very closest cell types, majority of the chromatin information are similar. Only in few places or few loci you will get difference, but majority will be the same. So the rationale is that once we identify this hotspot region, we will use this as a targeted region to probe more in the patients to see whether they have integration at this site and how do they progress over the time. Yeah, that's
Thank
it.
you.
Yeah.
Our next speaker is Dr.